Yes.
So thank you everyone for, thanks for the opportunity to talk to you today. So I am, if I can figure out how to change the slides. So my talk is really just going to be in three kind of parts. So antibody, I'm first going to talk about antibody germline genetics, then HIV specific antibody repertoires, and then where to from here. So antibodies protect us against many different infections. And so you have billions of antibodies within your system, which I'm trying to show in here. And so each of these antibodies are unique, which are represented by the different colors, but the antibodies are also antigen specific. So you have just showing it here with the colors. So this antibody is specific to this particular pathogen. So antibodies are essentially broken down in two different regions. So there's the Fab portion, which is what binds onto the antigen and it blocks infection through a process known as neutralization. And then there's the second part, that's the FC portion, and this is what is responsible for effector function. And that just means really different ways to kill the pathogen. And some examples of those are through ADCC, phagocytosis, and trogocytosis. So antibodies are made up of heavy chains shown in the gray here and light chains shown in black. But it's really, it takes 7 different types of antibody genes to make up a fully functional antibody. And so the heavy chain is made up of variable diversity joining and concentrating genes. The light chains have the same type of genes with the exception of the diversity. And so each of these antibodies have, or each of these, genes have numerous versions. And so each of those are essentially housed on 3 different loci in a human genome. So all of the heavy chain antibody genes are found in chromosome 14, and there are 2 different light chains, lambda and kappa, and they're each on different chromosomes. So antibodies come about through a process called somatic recombination, where essentially a single version of each of the V, D, and J's recombine to form the VDJ on the heavy chain, and then the V and J on the light chain. The heavy and light chains join together to form a fully functional antibody. So it's a little bit like building with Lego. So if you think about each of the different shapes of the blocks being the V, D, and J genes, and then the colors are different versions of those genes, and eventually you end up with slightly different towers. So germline antibody repertoire is highly diverse. So we know that there are single nucleotide variations within each of these genes, but we also know that there are large copy number variations within genes, whole genes as well. So Corey has shown this quite a few times. This is an example of a comparison to the reference where you can have whole gene duplications. And so individuals may have different repertoires, but even within an individual, you can have different repertoires on different chromosomes. So we've done some work on this in individuals within South Africa, and I'm going to talk a little bit more about the cohort a little bit later. But this is work that is done again in collaboration with Corey Watson, and it's been done by a student of mine, Elaine Marsden. I'm just going to focus right now on the heavy chain and zoom into a part of the heavy chain. So this, what I'm showing you here is a representation of 2 different donors. Each of the rows here represent different chromosomes, and each of the columns represent different genes. So just a zoomed in part of the heavy chain. So each of the gray blocks here mean that that particular donor had that gene. And if the block is in white, then they don't have the gene. So you can see that between these individuals, you can have very big differences in copy number, where whole genes are missing for some individuals, but also between chromosomes within the same individual. So we also see differences in alleles for each of these genes. So each of these colors represent a different allele, and the ones that are shown in red here are new or undescribed alleles that had been now described in this African population. But this is not specific to South Africa. We've seen this in other populations as well. So, and now it's not working. So up until now, I've been talking about diversity in the antigen binding portion, but we're also interested in genetic diversity in the FC part of antibodies. So the FC part is really what decides or determines antibody isotypes, and there are multiple isotypes shown here. Majority of these are seen in monomeric form, but IgM are seen in pentamers, and then IgAs can be seen also in dimers. So we focus primarily on IgG and IgA antibodies because our lab studies viral infections, and these are the most important antibodies within viral infections. So we've started looking at genetic diversity, and this is really a pilot study that we did with very few donors, roughly 10 donors, looking at genetic diversity in IgG1 and IgG3, and this is also work done by students of ours, Ty and Holly. And so IgG1 and IgG3 are fairly well described isotypes, but despite that, and despite small numbers, we identified 2 undescribed alleles within IgG1 and another 5 within IgG3. So the 2 alleles within IgG1 had synonymous changes within encoding regions, whereas of the IgG3s, at least 3 of these had non-synonymous changes resulting in obviously amino acid changes in coding regions, and this could impact function. And so we have some studies that are looking at the impact of these amino acid changes on function, but I'm not going to talk about that today. So we've also looked at genetic diversity within IgA1 and A2 antibodies, also within a small number of donors, and this is work done by Ty. And again, so IgA antibodies are less well described compared to IgG, and this is why we've identified far more alleles, probably in IgA, at least 8 undescribed alleles, and for IgA2, at least 5. And again, within these, we are seeing non-synonymous amino acid changes in coding regions that could impact function, and we have some studies looking at that, and then the same for IgA2. But how does all of this genetic diversity really translate to antibody responses to disease? So as you all know, COVID-19 has been a massive pandemic that we've been in for the last 2 years. And it's roughly accounts for over 5 million deaths, with at least 3.2 million deaths occurring in 2021. So this graph shows the number of deaths by month since the start of the pandemic in January 2020, or I guess December 2019, up until November, and they're broken down by geographic region. And you can see that Africa has relatively low reported numbers of deaths as a result of COVID-19. And this is in major contrast to what we see with HIV/AIDS. So this is the same graph, but now by deaths due to HIV and AIDS over the years since 1990. So HIV has accounted for 36 million deaths over the 40 years that we've had HIV, and you can see in red, Africa really bears the burden of HIV. And for this reason, I'm going to focus primarily on HIV in my talk today, but I am going to touch a little bit on COVID a little bit later. So the sub-Saharan Africa really has the highest burden of HIV, and South Africa, where I am based, has a particularly high burden of HIV cases. We also see it disproportionately affecting young women in their early 30s compared to men. And so we have partnered with Caprisa for many years now, and they really established clinical sites in parts of South Africa, in KwaZulu-Natal, specifically to address the issue of high prevalence of HIV in young women. And so the 2 sites that we mostly study are in Durban along the coastline, shown at the bottom here, and then Vulindlela, which is a rural area further inland within KwaZulu-Natal. So during HIV infection, everybody mounts antibodies, and these we call strain-specific antibodies. But in about 25% of people, we see the development of broadly neutralizing antibodies. And this essentially means antibodies capable of neutralizing globally circulating strains. And so we use the information from broadly neutralizing antibodies in a vaccine setting, and you can do this in 2 ways. And the first is through passive vaccination. And that really means is that we take these broadly neutralizing antibodies and we actually give them to people that in the hope that they would be protected should they come across HIV. And so we have some studies working on this, specifically using CAP256 VRC26, which is a broadly neutralizing antibody that was isolated from one of the capresa donors from the cohorts that I just showed you. But I'm not going to focus on that today. The second way of doing this is through active vaccination. And so the idea is that we take elite neutralizers, which occurs in roughly 5% of people. And these are broadly neutralizing antibodies that are really potent and really broad, and that you try and make this in a vaccine setting. So it's really, really difficult to do. And the reason why it's difficult to do is because it takes years of infection to develop these kinds of antibodies. And this happens when somebody becomes infected with HIV, their UCA, so they have an antibody response to this virus, but then the virus eventually will mutate from that. During this process, the antibodies will accumulate mutations through somatic hypermutation that allows them to neutralize new viral variants that are circulating. And so this process goes on. It's kind of like an arms race that happens between the antibody and the virus. And eventually, after many years, you are left with a broadly neutralizing antibody. So we use this information really as a way of providing a roadmap for vaccine design. And one way of doing this is using this information in a sequential vaccination setting. And I'll talk a bit more about that a little bit later. I wanted to just quickly touch on variability or genetic diversity within the virus. So this is an example now of SARS-CoV-2 genetic variation. So each of these little squares here, colored blocks, represents an amino acid change away from the original Wuhan strain. So some of them, you can see there's a lot of toggling, and some of them kind of stick because they have an added advantage. And this is specific to the alpha variant. And so you can see we start seeing persistent genetic variation within the virus. This is fairly low compared to what we see with HIV. So this is the same plot looking at genetic diversity within globally circulating HIV strains. So SARS-CoV-2 is an acute infection, and so the virus is cleared really quickly within weeks, and so we see really low variability within a donor. And this is in contrast to HIV, which is a chronic infection. So the virus persists for many years and people don't naturally clear the virus. So within one individual, you can have over the years of infection, their viruses can look a little bit like this, and we call this a quasi-species or a viral swarm. So I just wanted to make the point, it's important to think about this when I talk about coevolution, that we really are dealing with a high number of highly variable viruses within a donor. So today I'm going to talk about 3 coevolution stories from 3 different capresa donors, each with different levels of neutralization breadth. So for each of these donors, we've been studying their infection over years. We've also isolated antibodies from these donors, and we use the information from the isolated antibodies to amplify the antibodies over time. So we design primers specifically for their germline antibodies. And then we use the information between the viruses and the antibody to try and understand what the coevolutionary pathway happened within these donors. So for each of these donors and for many other coevolution stories that have been done, we see with the increase in infection, you get this increase in somatic hypermutation, so the accumulation of mutations within the Fab region. So the accumulation of mutations isn't always the same between the heavy and light chains. It really depends on which of the chains makes contact. So more often than not, it's the heavy chain that is more mutated than light chains. We also know that with increased somatic hypermutation, we get increased neutralization of viruses. So what I'm showing here going down the plot is just representatives of an antibody lineage with increased somatic hypermutation going down. and then different viruses that we've picked over time. And what you will see going down and across the graph is that antibodies, so this, is a heat map that shows the ability of these antibodies to neutralize the viruses. So the gray means that the antibodies can't neutralize the virus, and the warmer the color means that they're more potent. So going down and across, you'll see that the more mutated of antibodies can neutralize more viruses, but it can also neutralize it more potently. So sometimes this increase in somatic hypermutation results in neutralization breadth of global HIV strains, but more often than not, it remains strain specific. So not all mutations are beneficial. So we've shown this before with CAP256, but it's true for most co-evolution stories. So this is really the ideal pathway. So you start with a common ancestor, and the antibodies evolve to become broadly neutralizing antibodies. But sometimes what happens is that part of the antibody lineage or sublineages within there accumulate some mutations, but despite that, they can't neutralize new viral variants. And so those sublineages result in dead-end lineages that no longer continue to evolve. We see some that have high levels of mutations, but they don't become broad. So we call these off-track antibodies. And so while our ideal setting is to understand or to get to this path, it's really important to understand what is happening in these two regions as well, to know what not to do in a vaccine setting. So I said at the beginning, how does germline antibodies, or how do germline genes help with disease responses? So some germline genes predetermine your antigen target. This is especially if there are particular motifs within the germline that are important for binding. And these antibodies essentially form what we call public antibody classes. And this is kind of what I'm showing here. So this is an overlay of different antibodies from 5 different donors targeting the CD4 binding site. And you can see that they all essentially look the same. They have the same germline genes, they target the same thing. These all form within what we call VRCO1 class of antibodies. So there are multiple antibody classes for broadly neutralizing antibodies. VRCO1 targeting CD4 binding site is just one of these. But essentially, we use this information in a germline targeting immunogen design. So we use the information that we get from a co-evolution study, and we design the immunogens based on the viruses that engage with intermediates within a broadly neutralizing lineage. So the germline, you start with a germline targeting immunogen that engages with a precursor of a bNAb lineage, and then you give people sequential vaccinations based on different immunogens that engage intermediates of the lineage, with the idea of trying to provide or shepherd the immune system to create a broadly neutralizing antibody. And this isn't working again. So an example of this is, as I showed you before, VRCO1, which uses VH1-2 and it has a short CDRL3. And then EOD-GT8 is the immunogen that, the germline targeting immunogen that initially engaged VRCO1. So there are many trials that are ongoing to do this kind of vaccine design. So as I said, there are many different types of bNAb classes. And one of the classes that we have been looking at is an MPER class. So this is CAP206, CHARV12, VRC42, and 4E10 all target the membrane proximal external region, which is at the base of the envelope here next to the viral membrane. All 3 of these antibodies use a VH169 and a kappa 320. So VRC42 and 4E10, they also share a motif within the CDRH3, which we know is important for neutralization. So while VRC42 and 4E10 are amongst the broadest and potent of broadly neutralizing antibodies that we have, CHARV12 is an intermediate neutralizer. And so we used co-evolution of the CHARV12 lineage to try and understand the differences between these lineages. So this is just a phylogenetic tree of the heavy chain of CAP206 CHARV12, and you can see it sits here in the orange branch. But a lot of the continued evolution sort of happens between these two branches. And what that means, or what we saw really, is also convergent evolution. So what that means is evolution towards the same amino acids from different lineages in the same epitope. So all three of these lineages mutated towards this particular motif within the CDH3. So this was really exciting for us, and we thought maybe one of these antibodies might also be broad and potent, like VRC 42 and 4E10. So we tested these antibodies, or representatives of these antibodies, against a panel of viruses, and indeed we do get an increase in neutralization and potency with a red antibodies, and those are the ones that have the important motif. And in fact, if we mutate out the GL just of cap206-Trv12 to a GW, that single amino acid, we get significantly improved neutralization and potency. However, despite this, we still don't reach levels that we see with VRC 42 and 4E10. So because these antibodies all target the same part of the virus, we thought it might have something to do with the interaction with the antibodies with the virus. So what we did then is to mutate out this part of the virus, the part of the MPER that all of these antibodies bind to. So we mutate these individual residues, and then we test the ability of those antibodies to neutralize these different viruses. So what you're seeing here is a heat map of the fold change in neutralization, where the red means that the antibody can no longer neutralize the virus, where they could neutralize this, the wild type version. And so what is striking is that you see very different patterns between cap206-Trv12 and the GW version and the native version compared to what we see in VRC 42 and 4E10. So we modeled these amino acid residues onto the MPER structure, and you will see that VRC 42 and 4E10 target the hydrophobic core within the internal side of an MPER, whereas Trv12 targets the external solvent exposed side. Then when you compare the amino acid residues or the variability of those residues in global strains, these pie charts show how variable those particular residues are based on globally circulating HIV strains. You will see that this particular residue 674 is highly variable in globally circulating strains compared to these that are fairly conserved. So we think that it's because Trv12 is binding to the same, although the same epitope, it has a different orientation and it's dependent on highly variable residue, and this is why it can't become broad. So although germline genes are really important for BNAV classes, they're not always the only thing. So we've also learned from cap88-Trv06 is that regions outside of the Fab can also influence maturation. So cap88-Trv06 is an IgA isotype, and because of this, we were able to track isotype switching within the coevolution. So within this particular lineage, we identified multiple antibodies that have identical Fabs but different isotypes, and this allowed us to test the influence, the direct influence of the Fc portion or the isotype on antibody neutralization. So I'm just showing cap88-Trv06 here as an example, but we have multiple examples throughout the lineage of this. And so we made this antibody in the 3 different isotypes, and then we tested these antibodies to neutralize a panel of viruses. And what you will see is that we have very different neutralization profiles between these different isotypes. And this is a reminder that they have exactly the same Fab. So the difference is really influenced by the isotype here. And in this particular case, the IgA was the most potent and IgG1 is the least potent. Because class switching happens in a very specific order, we were able to then infer the influence of isotype switching on maturation within this lineage. So the initial precursor would have started off as an IgM switched to an IgG3, and this IgG3 version was able to neutralize the early precursor of the virus as well as a new viral variant with an escape mutation. We saw increased somatic hypermutation within this lineage, within IgG3, but then also class switch to different antibody classes. In this case, the class switch resulted in reduced neutralization of circulating viral variants, and all of these antibody classes then could resulted being dead-end sublineages. However, the IgG3 version of this lineage continued to evolve, accumulating somatic hypermutation, enabling it to neutralize new viral variants with escape mutations. But again, a class switch to an IgG1 resulted in reduced neutralization. Interestingly, though, we saw another class switch from an IgG1 to an IgA1, and in this case, the class switch restored neutralization back to what we had seen with the IgG3. And so this is something that we termed switch redemption, which is the redemption or the restoration of neutralization following a bad isotype switch or class switch. So the class switch to an IgA1 then enabled this lineage to continue to mature and accumulate somatic hypermutation and neutralize circulating variants. So this really challenged the dogma of, up until this point, it was always assumed that it was really just the Fab portion that contributed towards antibody maturation. We're able to show that isotype also can contribute towards this. So also up until this point, I've been talking about tracking antibody lineages or a single antibody lineage within a donor. But individuals mount multiple antibody responses to HIV and other infections. And so in this particular donor, we tracked 3 different lineages, two of which target the CD4 binding site and one glycan on the V3. They all have different germline genes. So despite increased neutralization breadth with increased somatic hypermutation, none of these lineages individually could neutralize all of the viruses that are neutralized by this particular donor. It's only when you add all 3 of these lineages together were we able to get the same level of neutralization breadth seen in this particular donor. So I just wanted to quickly conclude the first 2 chapters is that African antibody genes are highly diverse. We know that coevolution studies provide a framework for germline targeting vaccines, that certain germline genes can predetermine the target epitope, but it's not one size fits all. So we know now that orientation matters of the antibody and the virus. We know that the Fc matters, and we also know that polyclonality matters. So where do we go from here? So it's an iReceptor series talk, and so coming to an iReceptor near you, the NICD is creating their own remote repository. This will go online hopefully very soon. So all of these coevolutionary studies that I've been talking about will be on here, available for everyone to look at. We're also expanding our germline sequencing in collaboration with Corey, increasing our numbers of donors and also extending this to the whole IGH, the kappa and the lambda loci. We're also in the process of linking germline repertoires to the naive expressed repertoire. So we have matched germline data with naive expressed repertoire, and the question here is how do diverse germline genes influence antibody responses. So I'm just going to very briefly talk about SARS-CoV-2. So like many researchers, we've also had to pivot to understanding SARS-CoV-2, and I've been actively involved in SARS-CoV-2 surveillance. This really just tracks the number of genomes that we've sequenced from South Africa's pandemic, looking at the different variants that have been circulating. But we've also been actively involved in understanding antibody responses to these variants. So here I'm just showing the plasma neutralization response from donors that were infected with the initial D614G infection and their ability to neutralize the virus that they were infected with. We then compare this to their ability to neutralize circulating variants. And so we see a drop in neutralization as a result of immune escape by these variants. We've done the same with beta and delta infections, and what you can see is very different antibody responses depending on what antibody or what virus you have been infected with. So like HIV, SARS-CoV-2 also has antibody classes. And so I'm just showing here 4 different classes of antibodies that target the RBD. We know that there is also biases in germline gene usage, particularly with class 1 using VH353. But there is a specific motif within the CDOH2 that is seen in other antibody genes that we know are important for SARS-CoV-2 antibodies. But all of this information is really specific to 614G responses. So we're in the process now of trying to understand the differences in responses to different variants and the germline genes that might be contributing to these. So this is a massive effort. I've listed quite a few people here, but it's really a lot more people than are listed here. But I just wanted to show you this is our lab right now. It's headed up by Professor Penny Moore. It used to be headed up by Lynn Morris. She's still actively involved with us, but is now the Deputy Vice Chancellor of Research and Development at WITS. And then lots of collaboration across the board. And that is it.
Thank you so much for this very nice talk. So much data.
Too much.
Do we have questions from the audience?
I got a couple. Thanks a lot, Catherine, and it's so exciting that we're going to have a repository up in Africa soon. That's so cool. I mean, a couple. So, because you think about this stuff, pretty naive questions. So, I mean, you mentioned how there's very little variation within individuals for the, COVID antibodies, but I mean, is it so little that it's not useful? I mean, you never see, are they escape mutants, the little variability that's there, and is it worth looking at, or?
Yeah, so what we have seen in multiple occasions now is really in patients that are immunocompromised or have a long COVID infection that don't clear the virus very quickly. They do tend to generate viral escape mutations. And so there is some work ongoing looking at longitudinal viral sequencing of patients that have been infected for a long term. But we don't, because it's generally an acute infection, we don't see as high a variability as what we see in HIV. But it does happen. It certainly does happen. One of the things that I'm kind of keen on doing is applying a co-evolution story to COVID, like we do with HIV.
Yeah, that would be great, because it just seems like it's not even reported yet within individual variation, but it seems like it.
There have been a couple of studies that have come out specifically, I think in for cancer patients, and then in patients that were infected with HIV as well.
I was wondering that Africa deaths lower. I mean, that's probably partly just our bias in what reporting we see, but I hadn't seen that story that the deaths. I mean, it was everybody kind of in the background. You're wondering why the death. So is it the infection rate is super low, or the death rate is super low, or is it what?
So I think there's partly some of it could be a reporting issue, but generally we are seeing a lot less deaths than what we're seeing in other parts. And I think there's a lot of research ongoing to try and figure out what that might be. I don't have the answers for that. Yeah,
but I mean that figure you showed, it was, I mean, it wasn't like it was very super striking. And then this is kind of, but I mean, reading the SARS literature, it does they start talking about broadly neutralizing antibodies. I mean, do you think it reminds me of all the HIV work, and you think that's what's going to start happening, these targeted vaccines, and trying to that's kind of.
I mean, the germline targeting vaccine design.
I mean, it hasn't really.
So there's a lot of work ongoing, particularly around the VRC01 and EOGT8. But people are looking into other BNAb classes as well. It's really specific to a BNAb class, because the idea is to try and get engaged sort of a public antibody type. But I think also part of the reason why I showed the difference in diversity between SARS-CoV-2 and HIV is just the massive challenge that it has been for us to develop a vaccine for HIV. It's just that much harder. HIV is far more diverse. People don't generally clear it. It's a chronic infection. So, but I think we have learned a lot of lessons from SARS-CoV-2 vaccine development, and probably a lot of those lessons would be translated and reinvigorated, I suppose, into the HIV vaccine design. But definitely, I mean, germline targeting is a way, is one avenue that people are going and looking at sequential vaccination based on different viral immunogens that we see in coevolution. Of course, other ways of doing this, and the reason why I brought up polyclonality is because in a passive vaccine setting, the field generally, the idea is to give multiple broadly neutralizing antibodies and not just one. And with the, the idea of trying to target the virus from different angles to try and minimize viral escape. So that's, we are also looking into trying to use multiple antibodies as well.
I got one more of that, but with everybody's focused so much on COVID, are there HIV vaccine trials that are, going on that we should know about? It's a stupid question too, but.
So there are trials. There are plenty trials that are going on. For HIV vaccine, I think it has in a way not stagnated, but it has, our focus has changed a little bit, because a lot of, at least initially in the COVID pandemic, is that a lot of HIV researchers have had to pivot to SARS-CoV-2, and a lot of that is because the assays that we use to understand neutralization all comes off of HIV. It was, in a way, we had set up a lot of these things, and it was easy for us to translate that into trying to understand SARS-CoV-2 research. So many HIV researchers have moved to SARS-CoV-2, but we're also trying to get back into HIV. And so we're trying to do a little bit of both, but the HIV research is still ongoing. In South Africa, we're trying to get back to it because it is such a major burden for us.
Yeah, it just seems that iReceptor Plus, if we could get in, we really don't work with a lot of people doing clinical trials, but it would seem like such a, the iReceptor Plus has a lot of resources that could be, effectively used by people doing clinical trials. What we typically work with just almost, academics that aren't actually doing the trials. But, it might be, these ongoing HIV vaccine trials might be a place to try to make some contact.
Yeah, I think also that was another reason why we kind of opted, to establish a repertoire or a repository for this, to try and engage other HIV, researchers to deposit HIV sequences in there. So we will be depositing, our data, obviously, and also data from other collaborators. So eventually CAP256 data will be in there. We also have bulk sequencing that we've done in collaboration with Evelyn Georgiev, looking at public antibodies. So that data will be in there. And I think, iReceptor is really an extremely powerful tool for us to identify, public antibodies, and particularly if we're aiming for germline targeting, this is an incredible resource. So I guess it's a plug to also, get people to kind of use it and deposit data. If not in our repository, it's not going to be an HIV specific, because eventually we will add some COVID things in there, but to really just deposit data in there just makes it more powerful.
Yeah, thanks. Yeah.
Any more questions for Cathrine?
Yes, please. It's William. Hi, Kathy. It's great.
Hi, William. I haven't seen you in so long.
Yeah, I know. I know that another approach that's been taken is to present multiple antigens at once. And I wondered how you contrast the two, and whether you thought the class switching discovery influenced which one might be better.
So the multiple antigen at once, it is a great idea. Well, in my opinion, I don't know as much about it as I do about germline targeting, so I can only give my opinion, and please don't take it as the gospel truth, but in my mind would be a good approach, because that's kind of what happens in natural infection scenario, is that you're bombarded with multiple circulating variants. And you don't mount a single antibody response. People mount multiple responses. We've seen, polyclonality can contribute overall breadth. If the virus is escaping from a single response, then you have other responses that you can then, to try and take over. So I like the idea of it. I haven't seen much data on it, but that's not to say that there isn't data out there. There probably is. In terms of the class switching, so I don't know how that, one way of dealing with the class switching in a scenario is using adjuvants. So we know that particular adjuvants can induce particular class switches, or they can result in broadly in class switching. So it depends on, one of the things that I would like to do is to just trial a whole bunch of different adjuvants and see if we can in a way force a class switch, not a specific one, but generally class switching, and see if that helps. I think at this point, anything will help. HIV vaccine research, we've been doing this for 30 years. So I think that's also why HIV research has pivoted, and we're excited also about SARS-CoV-2, because, we got a vaccine pretty quickly in some ways, and it's really exciting after years of trials and nothing. So this has been great in that regard.
Well, thank you.
Any more questions? Then I have two questions. So you mentioned in a very brief remark that you saw evidence, I think it was one of the earlier slides, that you said that SHM rate correlates with those amino acids that are contacting the antigen. Is that something that you see in general, or?
Yeah, so in general, what we see, so how we start off normally with a co-evolution story is that we go in, it has to be dependent on a particular antibody that we isolate. Then we go in targeting their heavy chain and light chains. And then we look at just in general, what level of somatic hypermutation are we seeing in the heavy chain versus the light chain? If it's fairly even and you see somatic hypermutation happening in both fairly evenly, then we know that at least both of the chains are making contact at some point. We don't know where those contacts are. But a lot of the time we see, so for CAP88, TAVIO6, for example, we see more somatic hypermutation definitely in the heavy chain and the light chain, and then eventually we figure out that actually the contacts in the heavy chain. So this can be your first kind of indicator if you're seeing more somatic hypermutation, one of the chains, or if it's even, then you know that both are really playing a role in contact before you have a structure. Structure will obviously tell you that as well, but sometimes that is not as easy to get as just sequencing the repertoire.
Very nice. And then a more theoretical question. So you showed that some germline genes are really important for certain epitope targeting. And we also know, for example, from flu that some germline genes are important, right? And so how, and this is for some antigens, right? And ideally, we would like to know how the true relationship is between germline genes and pathogens over evolutionary time, let's say. So is there, do you know of any evidence, or are there studies where people have looked at or starting to look at, how have germline genes evolved over time by helping us to respond maybe quicker to certain pathogens or something like that? Yeah. Giving us a starting point.
Yeah, it's an interesting question. I wonder if that's something that Corey has seen before, because this is something that he kind of looks at all the time, or has been, I think, more so than most of us has a better handle on what the germline, human germline repertoire looks like. But I don't know of studies that do that, and it's an interesting question to see, how they would have evolved over time. We definitely know, we initially had a reference, and we thought we knew all the genes that were there. And actually, the more we look, I mean, you know this as well. The more we look in different populations, we're seeing not just different alleles, but different copy number.
Yeah.
You know, different genes altogether. So I think we really don't have a good handle on the human germline repertoire. And, you know, that's really important to also understand, you know, from different populations. So, yeah, that's an interesting, we've always just thought like, how, you know, how does germline impact the actual expressed repertoire, but how is the germline changing? I don't know. Interesting, yeah.
Thank you. Are there more questions?
I was just going to say about that germline changing. I mean, there are these well-known like somatic mutations in flu. You know, this, I don't know, 323 always has to take on this particular, and you know, you go, why doesn't it get coded into the germline? You know, but there's these repeated somatic mutations that, you know, it's a strange, it's an interesting question. Yeah.